Hi, thank you for the introduction. Can everyone hear me in the back? Okay. My ears are blocked, so remind me and raise your hand if you can't hear me. So, as Kirk said, I am an assistant professor at UC Merced, and whenever I tell people that, they always ask me, where is UC Merced? So I'm just going to take the next few seconds to tell you where it is. So this is California, and UC Merced is about right here on the east side of the Central Valley, and it's about an hour away from Yosemite. And I used to say that I lived in Ithaca, and I used to say that Ithaca is 4 hours away from anything, but UC Merced is 2 hours away from everything. So everything on this side is 2 hours. Okay. So one of the things I'm really interested in is human adaptation. And so what this map is showing you is some of the estimates of human migrations. So now from the fossil record and also from genetic data, we know that humans originated in Africa about 200,000 years ago. But eventually they moved to other places within Africa. They moved into Europe, into Asia, into Australia, and all the way to the Americas. And so in doing so, they encounter many different environments, such as changes in temperature, changes in diet, changes in pathogen exposure, changes in altitude. And so the idea is that these different environments likely acted as selective agents that led to many local adaptations. And so there's a huge amount of just phenotypically, humans are very different. And so it's been one of the big questions in evolution is whether, what are the relative contributions, say, of adaptation versus genetic drift? And what is actually, why do we see so much phenotypic variation? And what is causing that? And also, just to keep motivating natural selection, it's not just interesting in terms of evolution, but it's also interested in terms of function. If you find a region that, say, it has been targeted by positive selection, then it likely affects a phenotype, so it likely affects function. It's interesting also in terms of disease. A lot of people are interested in understanding what's the distribution of selective effects of deleterious mutations, and Kirk's going to tell you more about that in his talk. And just to give you an outline now, I'm going to do a combination of tutorial and research talk. And so first I'll show you some introductory slides, and then I'll talk a little bit about selective sweeps and selection on standing variation. These are two models that have been widely used to study human adaptation. And then I'm going to tell you also about selection on introgressed DNA. And here, introgressed from Neanderthals and Denisovans, which are archaic humans. Okay, so so first I want to tell you about a summary that has been used a lot, not just to infer natural selection, but to infer demography, and that's called the site frequency spectrum. And if you read papers on population genetics, you will see that a lot in the literature. And I'm just going to quickly go over what summary that is. So if you have a set of sequences, let's say these are human sequences. So the rows are sequences, and each column is a position in that sequence. You could be, one way to summarize the data is to look at the polymorphic sites, or the sites that are variable. And the other thing you can do is align these sequences to the chimp sequence and ask, what is the frequency of the derived allele? The derived allele is the allele that's different from the chimp. So in the first column, everyone has an A, so there's no difference there. In the second column, you have two T's, and those are different from the chimp sequence. So you would say the derived allele is a frequency 2 out of 6 in this case. and so on. And so with that, you can generate a sequence of numbers, and then you can summarize the sequence of numbers. You can just build a histogram. So if you build a histogram, this is what it looks like. There are 10 sites where the derived allele frequency is 1, and so on. And that's what people call the site frequency spectrum. Okay? And so, so this is a summary of the data that you can use, but then you need to know what to expect. What do you expect? So we can use theoretical models or models of evolution to get some expectations of that summary of the data. And so one of the models is neutrality, meaning that mutations are neutral. They are not deleterious or beneficial. And so the model is very simple. You have a population, and everyone has a blue allele. But then at some time, an individual gets an allele, a red allele, a new mutation. And then by chance, that mutation can increase in frequency. But also by chance, that mutation can decrease in frequency. You can have a model of positive selection, where again you have your population, you have your new mutation, but in this case it's a beneficial mutation, so that individual has a fitness advantage, and so they're likely to have more kids, and their kids are more likely to have more kids. So over time, this might go to high frequency in the population. You can also have negative selection, where you have your new mutation, but the individual that carries that mutation now has a fitness disadvantage because it's deleterious. So negative selection is going to act against that mutation and remove it from the population. And so to model this, Jerome last week talked to you about the Wright-Fisher model. And that's how you can model the frequency trajectories of these mutations. So again, just a reminder, if you have a population of individuals at time t, and you have an individual with a new mutation, you can model the number of individuals that carry that mutation at any time t. And to generate the next population at time t plus 1, you simply just sample with replacement. So this is just xt is a Markov chain, and the transition probabilities are binomial. And so under this model, you can either get analytical expressions, but here I'm just going to show you a simulation. So here we have generations on the x-axis and the frequency of the mutation on the y-axis. And here you can simulate. You can think that mutations arrive at every time point, and you can follow the frequency trajectories of these mutations. And as you can see, these are neutral mutations, so most of the trajectories actually don't reach very high frequencies. Most of them either enter the population and then die out, or stay at very low frequencies. But there are a few trajectories that actually increase to high frequencies. And if you think of taking a snapshot at some point in time, and if you ask at that slice of time what's the distribution of allele frequencies, it turns out that it looks like 1 over x. So that's for neutral mutations. And in your data, you have a sample. So here you can ask, well, what does the site frequency spectrum look for a sample, say, of size 10 or 20 chromosomes? Under neutrality, well, it looks like this. So you see that most of the alleles, most of mutations are actually rare, and that's because those trajectories almost never reach high frequency. They mostly stay at low frequencies. If you have positive selection, then you see a higher number of mutations that have high frequencies than the neutral model, and that's because those trajectories are going to be more likely to reach higher frequencies. And if you have negative selection, then you see that most of the negatively selected mutations are going to stay at low frequencies. And that's because selection is acting against them and weeding them out from the population. And there's other models you can think of, such as fluctuating selection, which instead of modeling selection constant in time, it varies over time. And just to show you one application of this, this is from a sample of 200 Danish individuals. If you wanted, we sequence the exomes, and we wanted to know, we wanted to look at their side frequency spectrum. So here, the green line is the side frequency spectrum for synonymous mutations. These are mutations that are more likely to be neutral. And as you can see, the neutral expectation looks like not such a bad fit to the data. And if you look at the side frequency spectrum of the non-synonymous mutations, these are mutations that are more likely to affect function because they change the protein sequence. You find that there's a higher proportion of mutations, of non-synonymous mutations that have low frequencies. And so this suggests that negative selection is a better model for the non-synonymous mutations. And it says that these mutations are more likely to be deleterious. And you can actually try to fit a distribution of the selective effects of these to the non-synonymous mutations. So for example, you can fit this distribution that says that a proportion of the non-synonymous mutations are neutral, and the rest are going to follow some distribution of selective effects. Here we chose a gamma, and so you can infer these parameters and try to infer the distribution of selective effects of the new non-synonymous mutations. But of course, Kirk's going to tell you more about that next time. Okay, but going back to positive selection, so this assumes this is positive selection on a de novo mutation, meaning that there was a new mutation that was immediately beneficial, and then it reached high frequency. And so under this model, many people have thought about what are the signatures that we can look for in the data to detect selection on a de novo mutation. And so if you have your sequences here, you can think of this sequence gets this new mutation, and because that mutation is beneficial, the haplotype that carries that mutation is going to go to high frequency. Okay, so if you group the haplotypes with that don't carry the beneficial mutation and the haplotypes that carry the beneficial mutation, then there's going to be a lot of haplotype similarity in this group, because if it's very, very recent selection, that haplotype is going to go up in frequency really, really fast. And there's not going to be a lot of time to accumulate mutations on this haplotype. So a lot of people look for signatures of either a really long haplotype or for a decreasing heterozygosity, because the variation at this locus will be very low relative to a non-selected locus. And so a lot of people have developed summary statistics to look at that. You can do it with the SFS, because under that model, not only will the beneficial mutation go to high frequency, but all the neutral mutations that are linked to it. So you can look for departures of neutrality with the side frequency spectrum. There's things like Tajima-C, which is just a summary of the SFS. You can also look at FST, because if there's positive selection in one population but not in another population, then you expect there to be large frequency differences. There's also a lot of haplotype-based statistics that measure haplotype homozygosity. So the longer it is, the more likely it is a recent sweep. And but most of these are signatures within a single population. The ones FST and XPEHH actually use two populations instead of one population. And so using these summary statistics, there's been a lot of regions that have been detected, such as the lactase persistence gene. This is a gene that, so most of us can digest lactose when we are little, but then when we become adults, we lose that ability, with the exception of European populations and some African populations. And so if you look at Northern Europeans, they have a very long haplotype that has a putative beneficial mutation at very high frequency that's not present in other populations, suggesting that they can still digest lactose. And so a slightly more complex model is selection on standing genetic variation. So rather than have selection occur immediately after the mutation arises, you can have this mutation can arrive in this population and then drift, because it could be a neutral mutation initially, and then drift in the population at low frequencies. But then, because the environment changes, it can actually become beneficial, and then it's going to go to high frequency in the population. So that's a slightly more complex model. And so so these are the two models. So this is the allele frequency of the beneficial mutation. This is time. So this is the present, and this is the past. And so if it's a new selection on a new mutation, then it's going to enter the population. It's going to go to high frequency really fast. But if it's selection on standing variation, the mutation is going to enter the population, drift at low frequencies, and then, because the environment changes, go to high frequency. And so these are actually the two models that have been widely studied in the literature. There's a lot of papers looking at these two models of positive selection and trying to detect signatures of these types of positive selection in human genomes. And selection on standing variation leaves a slightly different signature, because unlike the first case, there could be more than one haplotype that carries the beneficial mutation. So in this case, you have this haplotype and this haplotype. And when that mutation becomes beneficial, the two haplotypes are going to increase in frequency, not just one of the haplotypes. So this makes it slightly more difficult to identify this pattern, because some of the methods that depend on haplotype homozygosity are not going to be able to detect this. And there's actually a lot of data sets that have been used to detect human adaptation, or adaptation in humans. There's the Human Genome Diversity Panel data set. This is many populations, about 52 populations, but it's genotyping arrays, so it's not whole genome sequencing. But now there's a lot of new data sets that are being generated, such as the Simon's Diversity Project. This actually sequenced more than 260 whole genomes, and from 127 populations. I guess the number of individuals per population is not very high, which you need to detect selection. But there's also the 1,000 Genomes Project. This is also a public data set, and it's about 2,500 whole genomes. And of course, there's also archaic human DNA. So for a long time, we knew that Neanderthals and modern humans coexisted, but we didn't really know if they made it. But in 2010, the first draft of the human genome, of the Neanderthal genome, was sequenced from bones in Croatia. And now there's a second Neanderthal genome from the Denisova cave in the Altai Mountains in Siberia. And there they also found bones that were not from a Neanderthal individual, but from a Denisovan individual. And what's interesting is that there is no fossil record for the Denisovan, but now we have its genome, which is quite interesting. There's a lot of data too now on ancient modern humans. So humans, there's data from samples that are, the youngest is like 100 years old, and now the oldest is about 100,000 years old. So now when we look at positive selection, not only can we use the modern data, but we can also look at the ancient data, and so this brings a new temporal access to detecting positive selection. Okay, and then in this slide, I like this slide because it shows most of the regions that have been detected to be under positive selection. Not all of them, but most of them. For example, here's the LCT gene, the lactase persistence. There's a lot of them to do with malaria. Others to do with diet or exposure to cold climates. There's pigmentation genes. And but the ones that I've worked on are mostly on high altitude, and I'll tell you a little bit about that. Okay, so in case you didn't know, there's 3 regions of the world that are considered to be at high altitude, and that's the Tibetan Plateau in China, the Ethiopian highlands in Africa, and the Andes in South America. And these are very extreme in the sense that people live in regions of more than 4,000 meters in altitude. And just to give you a sense of what that means, so for every breath of air that you take, there's about 40% fewer oxygen molecules at that altitude than at sea level. So that's a huge reduction. And so initially, people started studying high altitude in the Andes when somebody noticed that when they travel from low to high altitude, their hemoglobin concentration would increase. And they asked, well, does that happen with high altitude natives? And it turned out that in the Andes, that's true. High altitude natives also have high increased hemoglobin concentration at high altitude. And so for decades, that was thought to be the beneficial response, but it wasn't until the 70s that studies in Tibet showed that they actually had a different physiological response. So here I'm showing you Tibetans and Han. So the Han are from low altitude and the Tibetans are from high altitude. This is altitude on the x-axis and hemoglobin concentration on the y. And what you find is that if you look at the Han, the hemoglobin concentration increases as altitude increases, which made sense. But in Tibetans, there's a slight increase, but then when you look at these two, they're quite different. And Tibetans do not have to increase their hemoglobin concentration nearly as much. And so this suggested that the Tibetans had a different physiological response. And this study and many other studies looking at the heritability led people to suggest that Tibetans were not just acclimated to live at high altitude, but that they were genetically adapted. And so what we set out to do was to identify the regions of the genome that could explain this, this difference. And so we sequenced some exomes from Tibetans, and we did a lot of stuff, but I'm just going to show you a plot that really just summarizes it. So this is a 2-dimensional psi frequency spectrum. Here you have the Tibetan frequency on the x-axis and the Han frequency on the y-axis. And what you see here is, and the different colors are just the different heights of the bins. And as you can see here, most of the stuff lies along the diagonal. suggesting that these two are highly, that these two populations have frequencies that are highly correlated. But there are some mutations that have high frequency in Tibet and low frequency in the Han. And if you just simply look where they are, these two mutations are in a gene called EPAS1. And EPAS1 is really interesting because it's a transcription factor that regulates the response to hypoxia. So we were happy because it's a gene that makes biological sense. A lot of the times when you have positive selection scans, you find something, but then you have to come up with the story because you don't know if it makes sense or not. But in this case, it makes sense. And if you take just this mutation, we also genotyped it in many individuals, and we also collected hemoglobin concentration for those individuals, and you see a significant genotype to phenotype association. And it turns out that the individuals that were homozygous for the frequency, the allele at high frequency, had lower hemoglobin concentration, which makes sense because Tibetans do not increase hemoglobin concentration. They deal with hypoxia in some other way. And at this gene also you see what you would expect under a model of local adaptation. So here you have FST on the x-axis and maximal allele frequency on the y-axis. And all the points are pairwise comparisons of 52 populations from different places in the world. So humans are very similar to each other, so you will never see an FST of, say, 1 genome-wide. The highest we see is about 0.2. So these populations are probably more differentiated because they're from very they're far away from each other. So these are probably something like Africans and Asians, or that would be my guess. And populations here are populations that are more genetically similar. And so for EPAS1, FST between Tibetans and Han is about 0.01, so it's about here. But in that gene, you find mutations that have very large frequency differences of higher than 0.8, and that's higher than you would expect if this is your expectation. So the only other genes that show such a striking signature are genes involved in skin pigmentation. Okay, but in evolutionary biology, we're also interested in understanding the history of the selective event. So we had identified an interesting candidate of positive selection. But other questions that are of interest in the field are, well, when did the selection occur, and what was the strength of selection? And to estimate parameters, you need a model. So if you look at the two models that were definitely the most are the most popular models are selection on standing variation or selection on a de novo mutation. And so this is also interesting because EPAS1 was not the only gene that we identified. There were other genes in the hypoxia pathway that we identified, and so we wanted to understand whether all the genes were selected at the same time, or whether it was or whether it was a series of events. And so we tried to distinguish between these two models using an approximate Bayesian approach, which basically means you do a lot of simulations to generate data under these two models. You summarize the data, you say, okay, let's look at the real data, and you see how you keep all the data sets that are similar to your observed data, and then you build a steered distribution of the parameters. So it turns out that neither of these two models was a good fit to the data. And so this is we resequenced the whole gene. For identifying the gene, we just used exome data, but the two mutations that we identified were in an intron. So we resequenced the whole gene and tried to fit these two models, and neither of them was a good fit, which was really disappointing. And it was disappointing because when you say look at FST at this gene between Tibetans and Han, so this is the position, and this is FST per SNP. You see that there's a lot of mutations that have very large FST. So these mutations are very differentiated in these two populations. And just as a reference, the genome-wide FST for these two populations is 0.01. But there's a lot of mutations that have FST of 0.8, which is really, really high. And so that was disappointing. And so we decided to look at the data a little bit more, just the raw data. And so I decided to focus just on this region because that's the region that has the largest, the mutations with the highest FST. And so here I'm just showing you the data as a heat map. So rows are the haplotypes. So first you have all the sequences for the Tibetan individuals, and then you have all the sequences for the Han individuals. And each column is a variable site, and if it's dark, that means that it's a derived allele. So that haplotype has the allele that's different from chimp. And if it's gray, that means that the allele is the same as the chimp. And as you can see here, there's a haplotype that's at high frequency in Tibet, and it's quite different than the haplotype in the Han. Okay? And when we saw this, to us, it was clear why neither selection on standing variation nor selection on a de novo mutation were good fits to the data. And let me just tell you why. So if you look at the first 5 columns, these are the first 5 SNPs. The sequence that's at high frequency in Tibet is the sequence that has all derived alleles. And that sequence, it's a very high frequency in Tibet, but it's not, you don't see it at all in the Han. And this is about 2.5 kb in sequence, which is not very large in terms of the length. And just from what we know about mutation rates in humans, it's very unlikely that 5 mutations arose on the same sequence and then went to high frequency in Tibetans. And so to us, this was a suggestion that this sequence did not originate in Tibetans, but it originated it came from another population. So there must have been an admixture event from another population into Tibetans, and they got the sequence from them. Okay? And so under this model of adaptive gene flow, so receiving a genetic contribution from another population that's beneficial in the recipient population would lead to this. So if, let's say this is the haplotype that's been donated to the recipient population, then if this is beneficial, this would go to high frequency. But a lot of the neutral mutations hypothesized to be under adaptive introgression, or examples of adaptive introgression, and most of them are genes involving immunity, skin pigmentation, and metabolism and altitude. And that sort of goes with the story that that's because these humans have been exposed to, say, different diets, different temperatures, and different altitudes, and different pathogens. And so, but what we found was that most of the approaches to identify examples of adaptive introgression either did one of two things. Either they first identify introgressed segments, and that's something that Shiram is going to tell you more about how to do that, and then apply test of positive selection, or identify selection first and then look for signs of introgression. And so the problem with that is that introgression alone could actually lead to false inferences of positive selection. And also, if there's a lot of positive selection, then that could bias your estimates of introgression proportions. And so what we wanted to do was to model both selection and introgression jointly to try to understand this adaptation. And initially, the idea was to, rather than just model this and simulate data to look at the patterns of genetic variation, we wanted to look at the data and ask what features of the data are informative about this type of process. And then once you have the features, then you can do lots of, then you can build a test. You can put it, you can use the features to, and a hidden Markov model framework to detect these regions like this. And so to learn about adaptive introgression, we decided to look at the EPAS1 example, and you can use both haplotype information or SNP frequency information. But what I'm going to show you, we're just going to use SNP frequency. So when people detect introgressed tracks in modern humans, that doesn't mean that those tracks are identical to what you see in modern humans. There's some differences between the introgressed tracks, between the archaic human sequence and the modern human sequence. And that's because a mixture happened a while ago, and since then, the modern humans have accumulated mutations on that sequence that are not going to be present in the archaic human sequence. And so a very simple question is, how many differences do you see between, say, the most common Tibetan haplotype and the Denisovan haplotype? And you see six such differences. So there are 6 differences that, well, that's not what I want to say. There are 6 differences, or 6 mutations, that are uniquely shared between the Denisovans and the Tibetans. And all the other mutations in that sequence are either in other populations or are present in Tibet and not present in the Denisovan. So even though the introgressed, the haplotype in Tibetans and the Denisovan haplotype has only 9 differences, there's only 6 of those sites have are unique, what we call uniquely shared mutations, meaning that they are only present in the Denisovans and in the Tibetans. Okay? So that's, and the question is that surprising? Is that a lot of, it doesn't seem like 6 mutations would be, 6 uniquely shared mutations would be very informative about adaptive introgression. And so what we decided to do is to ask, well, if you look at a bunch of genomes and you ask, if you just scan the genome and ask for windows of 40 kb, and we use windows of 40 kb because that's the average introgressed segment, how many uniquely shared mutations do you see for each population? So that's just saying how many mutations are in the Denisovan sequence or the Neanderthal sequence and in that human population, but not in anybody else. Well, these are the results. So these are the window, the window number, and then this is the number of uniquely shared mutations. And as you can see, most windows have, there's a lot of windows that have a bunch of uniquely shared mutations, like over 50. And in EPAS1, we only had 6. So that's probably not very informative. And you see that the X chromosome is the green thing here. They have a lower number than the autosomes, which there's some reason why that is, that Shriram's going to tell you about. So this wouldn't be very informative to just use the number of uniquely shared mutations, but you could ask, well, what if you also take into account the frequency of these mutations? And if you do that, then if you ask for every window, what's the number of uniquely shared mutations that are higher than, say, 50% in frequency, which is pretty high, then you actually get, then you identify all the genes that have already been identified. You identify all the genes that were in that table that I showed you. So using those two measures, you can actually recover that. So then one potential thing to do, which is very simple, is to say, okay, well, let's use both the number of uniquely shared mutations and say a summary of the frequency of these mutations. And here I'm just showing you the 95% frequency quantile. And if you do that, then you can you can see that there's a lot of this is in Europeans. You see a lot of other you recover the genes that have already been identified, but there's also other stuff that hasn't been identified already. And we did a lot of simulations to show that these two summary statistics have a lot of power to detect adaptive introgression. But here I'm just going to show you some results on how these two summary statistics behave in human globally in different human populations. So if you look at the number of uniquely shared mutations of any frequency, you find that South Asians have the largest number of mutations that are shared with with Neanderthal and also with Denisovan. So South Asians have the largest number. But if you ask who has not only the largest number, but the largest number at, say, with frequencies higher than 20%, then you see that East Asians have the largest number of mutations that are uniquely shared with the archaic humans, but also at frequencies bigger than the 20%. And interestingly, you find that Peruvians also have a high number of these mutations. If you go slightly higher, 50%, then you find that the population with the largest number of uniquely shared mutations at high frequency, that population is the Peruvians, but you still see that the East Asians have a large amount. And if you just look at the genome-wide quantile of these uniquely shared mutations, you see again that Peruvians have the largest number. And of course, Peruvians probably went through two bottlenecks, so that has a tendency to increase these frequencies of mutations. So that would be the most parsimonious explanation, that the Peruvians have this excess of high frequency uniquely shared mutations because of a bottleneck, but that's still yet to be determined. Okay, and so we use these two summaries to just tag these regions, but then of course what we wanted to do was to actually look at the data to make sure that those are indeed or correctly tag the regions that have been under adaptive introgression. And so here are some of the most extreme windows by continent. So these are the 9 frequency quantile for that window, and this is the number of uniquely shared sites. So BNC2, that's a gene that has been identified already. And if you look at the sequence patterns in that region, you see that there's at the very top, that's the archaic sequence, and then you have human sequences, the modern human sequences, and you can see that there's a haplotype that's a very high frequency in modern humans, and it's not very different from the archaic sequence. So in this case, there's about 20 differences between the archaic sequence and the modern sequences. And there's always, when you have adaptive introgression or introgression, you tend to see 2 groups of haplotypes. So here you see this group and this group. And Peruvians actually had the largest number of extreme windows, and I'm just going to show you the results for one gene that is an immunity gene that's also been identified in Papuans, which is the population that has the largest amount of denisovan admixture. And again, you see very nice similarity. And again, you see these two groups of haplotypes, one that's very close to the archaic sequence and one that is not. And this is a gene in Europeans. It's also an immunity gene. It's a lot of Europeans have a sequence that's almost identical to archaic humans. There's genes that have been associated with disease, with diabetes in Mexicans. But in general, there's a lot of examples that we have now, and these are genes that are either involved in immunity, metabolism, or in high altitude adaptation, which suggests that perhaps modern humans were allowed to, they adapted to these different environments thanks to receiving genetic contributions from these archaic hominins. And so with that, I just want to say that adaptive introgression may be more important than previously thought. And if you're interested, you can go check out this paper. And I would just like to thank my collaborators. Thank you. Any questions?
So do you have a sense of when the introgression into Tibet occurred with the Denisovans? Did it occur directly, or did it come from some other unsampled population?
Well, we are only inferring it through the Tibetan data, or assuming that it came through the ancestral population of Tibetans, and that's about 30,000 years ago. And it's more recent than the estimates of the Neanderthal introgression into modern humans, and consistent with what other people have identified.
So could it be multiple waves of introgression in with humans?
Some people argue that it is, that there's been one in South Asia, one in the ancestral population of Europeans and Asians, and one in Papuans. But I think some people say yes, some people say no, so it's still debated. Okay, thank you.
Actually, before you go, first of all, tea time is 3 o'clock, and then we have to redo every minute of 3 o'clock because we can't put the thing up full. So, yeah.
What?
First of all, 2 things. Can you stay and do the 3 more minutes in the middle of your talk?
Oh, okay.
Okay. And then is anyone else willing to like stay on that way? Stay in the audience, but that's totally off. No,
So neither selection on a de novo mutation nor selection on standing genetic variation could lead to these results. So our hypothesis was that the Tibetans acquired this sequence via introgression from another population. And so we set out to find what other populations had this haplotype, and humans are very similar, so we were expecting to see the sequence in other modern human populations. And as you can see, Tibetans have the sequence at very high frequency, but the rest of the modern human populations that we looked at had this yellow haplotype. And there were only 2 Han individuals that were heterozygous for the Tibetan sequence. And so this looks, for humans, this is very strange. And so I was talking to my office mate, and we were discussing the strange results, and he suggested that it was the Yeti. But we don't believe that doesn't exist. But that gave me the idea to look at the archaic humans. And when you look at the sequences of archaic humans, Neanderthals do not have this sequence, but the Denisovans actually have the sequence. which is really interesting. But if you look at the whole region, then you see here the two Denisovan haplotypes, the Tibetan haplotype, the modern human haplotype that's closest to the Tibetan haplotype at high frequency, and the Neanderthal haplotypes. And definitely the Tibetan haplotype at high frequency is most similar to the Denisovan sequence. And there's only 9 differences between those two sequences. And actually, Tibetans are not the only population that have this haplotype. There's been a study recently showing that where they sampled many different populations from the Himalayas, and they also have this haplotype. And so this was a new way of thinking, or not a new way, but a new. Some other people have already thought about adaptive introgression, but it was probably the best example, or one of the best examples that we had about adaptive introgression. And so these results led us to think, or to ask, well, what is the relative contribution of this type of process in human evolution? And also because it would be nice to see what other nice genetic gifts we got from archaic humans. And we were not the first ones to hypothesize that that adaptive introgression must have been important in human evolution. There were already a few examples that had been proposed to be adaptively introgressed, and most of these examples are genes involved in immunity, genes involved in skin pigmentation, genes involved in metabolism, and genes involved in altitude. And this suggested that the environments that likely acted as selective agents in these cases were different pathogens, different temperatures, different diets, and of course different altitudes. And so the problem too, when you look at these genes, what you find is that most of these genes were identified in one of two ways. Either first you identify introgressed segments, and then you apply tests of positive selection, or first you identify positive selection and then look for signs of introgression. And that's a problem because introgression alone could lead to false inferences.